His name is Dr. James Tour. He's a very famous scientist. He's a synthetic organic chemist. He received his Bachelor of Science degree in chemistry from Syracuse University. He has a PhD in synthetic organic and organometallic chemistry. I don't even know what that is, but we're going to find out. From Purdue University, and postdoctoral training in synthetic organic chemistry at the University of Wisconsin and Stanford University. After spending 11 years on the faculty of the Department of Chemistry and Biochemistry at the University of South Carolina, he joined the Center for Nanoscale Science and Technology at Rice University in 1999, and he's presently Professor of Chemistry, Professor of Computer Science, Professor of Materials Science, and Nanoengineering. Okay, and I'm not done yet. He has over 600 research publications, over 120 patents, with total citations over 69,000. That's on Google Scholar. He was inducted into the National Academy of Inventors in 2015. He was named among the 50 most influential scientists in the world today by thebestschools.org. In 2014, he's listed in the world's most influential scientific minds by Thomas Reuters, ScienceWatch.com in 2014. And he was named Scientist of the Year by R&D Magazine in 2013.
Well, how does a cell operate chemically? Well, this is a cell. It's utterly amazing. A cell is a machine. It is a factory. How do you get materials in a factory from point A to point B? You have these overhead automatic scaffolds that pick stuff up and deliver it. This is what a cell is a factory. We understand certain things about the cell, other things we don't. But, you know, if it has to transport material from one end of the cell to the other, you know how it does it? It will build a microtubule. It'll construct the microtubule and then transfer the material along the tubule. And then it deconstructs the tubule. You say, well, why doesn't it just leave it there? Because then the cell would become too rigid. You get too many. It just deconstructs it and rebuilds it where it needs to transfer. It's amazing. How does it do this? It's amazing. So I don't understand the vast chemical mechanisms in a cell, but it clearly operates. So it's not improper to ask the question, by what chemical mechanism does it function? The very question spawns further investigation. So generally, scientists are invited to ask questions. So the question often asked of me by students is, what do you think about evolution? Well, on my website is jmtour.com. And then if you go to the corner, there's a personal topics tab. And one of the bullets there is evolution creation. So I've written about this thing. So before we talk about evolution, let's first talk about the origin of life, because you have to have life before you can have evolution. Or how did life arise in the first place? You want to say, well, aliens brought it here. That's fine. I'm okay with that. But let's say origin of first life. Where did the alien life come from? Let's say, what is the origin of first life? And on Earth, life has to have 4 compounds. It has to have carbohydrates, nucleic acids, lipids, and proteins. Those 4 compounds. That's what life is made out of today. That's what you and me are made out of. We're not made out of silicon. We're not made out of tungsten. We're made out of these molecules: carbohydrates, nucleic acids, lipids, and proteins. So I just wrote this article this year on abiogenesis, or prebiotic chemistry, and yeah, that's supposed to be me. I mean, you write these articles for them, and then they I don't know where they get these artists, but they do this to everyone. And you know, I saw it, I was like, whoa. And then my kids saw it. They thought, they're you know, one of my kids, you see, there they go again, attacking the Jews. I said, no, they do this. They do this to everyone. Don't worry, just calm down. They do this to everyone who writes an article for them. But I just dissected origin of life. So I asked one of my very famous colleagues, National Academy member, I said, what's the origin of carbohydrates? He said, oh, it comes from formaldehyde in the foremost reaction. I said, okay, and he said, I'll send you an article. So I read the article by a guy named Eschenmoser, a great chemist. I've known of his work for many years. He's in his 90s now. And I just went to the synthetic protocols and I dissected it. So if you look at nature's molecules, so nature uses ribose. This is ribose, these 5 carbon sugars. Why do we want to focus in on ribose? Because the backbone of DNA is a carbohydrate. So you have the carbohydrate strand, and then the nucleic acids are hanging off. So before you can have DNA, you got to have the sugars and the carbohydrates, and you got to have the 5 carbon carbohydrate. So in trying to make this in a prebiotic world, all you can do is mix certain things, but you can't do real sophisticated chemistry because you're only supposed to use the molecules that nature had at the time. Things like formaldehyde, things like ammonia. And so he had to make these pentose sugars, but the pentose sugars have 3 stereogenic centers, which means that you're going to end up with a total of 8 possible isomers. You get these 4 pairs of enantiomers, and then between the pairs of enantiomers, they're diastereomers. So you don't just get one, you get all of them. So what happens is you need these 5 carbon sugars, or you need to have the D-ribose in particular. If it's for DNA, you have to now pull off one of the hydroxyl groups to be deoxyribose, unless you want to start with RNA, which is fine, and then you'd have ribose, but then it's a lot less stable. But the prebiotic system never knows that. So when we want to make something in the lab, we decide the target we want to make, and then we go ahead and make it. The prebiotic system doesn't know what it's going to make. Doesn't know. I think I'll build life today. No, it doesn't know that. There's no brain there in a prebiotic system. So what is the chemistry going toward? It has no target. That's a big problem. Try to make something without going toward it. You don't know what to go toward. It had to do this with all those classes of molecules. It never knew what to go toward. It never knew what to select because it didn't have a selector. It's not biology. Biology has selectors. Living systems have selectors. If it's the right thing, it notices it by this enzyme. If it's the wrong thing, it notices it and it sends another enzyme to chew it up. But this is before there's any enzymes because it's prebiotic. It's pre-biology. It's pre-life. Before you can have life, you got to have the molecules. So that was the first thing. This is there's a number of criteria that nature has to deal with. If you view this totally as a materialistic world, nature has chosen a hard route. It's chosen most of its molecules can be enantiomeric, and it's chosen them as homochiral. So the system has multiple stereogenic centers, and it's chosen homochiral. These are hard compounds to work with. Just trust me, it's very hard to do homochiral synthesis on something with multiple stereogenic centers. You have solubility problems. Try to do organic chemistry in water. That's what a natural system has to do. And you have to design the system so it splays out the hydrophilic portions and puts the hydrophobic portions on the interior. As organic chemists, we just use organic solvents. Nature's got to use just water. When building a molecular nanosystem, like trying to build a functioning cell, you have to do constant redesigns, because it doesn't work. So you go back to the beginning. Redesign often makes you go back to step 1 in the synthesis. I know this because we build nanomachines. Very few people build molecules all the way up to a nanomachine. Very few people. There's only a handful of us in the world which will start from molecules and go right up to a functioning machine. So it bothers people when I say I probably know more about this than most people, but let me explain to you why. There's so few people that do it. Even most organic chemists don't try to build a functioning machine from their system. They make a molecule. Hey, look at what I made. But they don't try to put them together to build a functioning system. That's what we do with these molecular cars and these submarines. The cars, by the way, have 4 independently rotating wheels. They have molecular motors. They have axles. They have loading bays, all of these things. But if you want to change something, you got to go back to step 1. How did this happen in a prebiotic world when there's no mind there? It doesn't know how to stop. So say it gets to a carbohydrate. How does it know to stop the synthesis? It doesn't know that, there's the target I want. No. You just ruined what you just took 400 million years to make. It doesn't know to stop. Prebiotic system doesn't have just-in-time delivery. I order chemicals, they're at my door in 18 hours. And I keep the intermediate safe and stored in the freezer for the next step. What does nature do? Here in this cave, something formed. The thing it needs to react with is in a cave 80 kilometers away or on another planet. How does it get there? How did it get there? It has to be in a concentration where things can react. If it's just one molecule in a puddle, there's not a concentration gradient where things can react because the 2 molecules never find each other. It's easy statistics to figure out. You have to have a certain concentration before things can react. People will often say to me, oh, if you wait long enough, it'll form. No, time is your enemy in organic chemistry. time is your enemy. You make something, you right away store it in a precious place because if air hits it or ammonia hits it, it falls apart. They're kinetic products. Very often they're the kinetic product, meaning that they form first, but they're not the most stable. The most stable is junk. Reagent addition order is critical. You have to add A, and then B, and then C. You can't just throw it all together. It doesn't work that way. It just doesn't work that way. Try cooking, an elaborate French dinner. I bought all this stuff. I just add it all together, and chemistry is the same way. You can't do that. How does nature know what to add? How does it add one and then the other and then the other? And when these guys do their prebiotic chemistry, say, look what we got. Yeah, because you added A and then B and then C, and you bought A and you bought B and you bought C. The parameters of temperature, pressure, solvent, light or no light, pH, oxygen, moisture, no moisture, has to be carefully controlled to build complex molecular structures. The characterization at each step is essential, but hard in a prebiotic system. How do you characterize it? Biology characterizes everything. Everything that's made, enzymes check it. And if it's not right, another enzyme comes and rips out that wrong base and fixes it in the DNA. But this is a prebiotic world. You don't have any of this. How do you bring up more starting material from the rear? Anytime you want to make a complex system, you go along and then you run out of starting material because you were trying many things, and then you go back and you make some more, and then you go back and how does nature go back? It just spent 1 billion years building something, and now, I ran out of starting material. Well, just go back and make some more. I would, but I never kept a laboratory notebook. I forgot what I had done 1 billion years ago. You see the problem. How many organic chemists are here? Okay, do you know what I'm talking about? Isn't this true? It just doesn't work. None of this works. And this is what they publish paper on after paper, and it gets into Nature and the Science. I'm like, you're crazy. And there's no accounting here. There's no accounting. So one guy makes ribose in a 0.1% yield with all the other carbohydrates in there, and they identify it by mass spec. And then the next researcher starts with ribose. Say, okay, ribose has been made, so we'll start with that. Now, no. Start with the junk that he made. You don't buy ribose with all the chiral centers in place that came from a natural source. None of it works. So critical for life is the origin of information. So this is just making the basic molecules. Now you got the whole information. It's not just the nucleic acids, it's the order in which they're arranged. The information is primary, the matter is secondary. Information is primary. You can write information on a piece of paper, then transfer it to your computer, put it up in the cloud, and now it's in some server form. The matter can change. The information is primary. This is why we believe that in Christ, when we die, we live forever with Him because the information is stored in the cloud. It is. It is retained in the cloud. We are very much with Him. The body can be cremated. The body decomposes. That's just the matter upon which it was stored for that time. And in fact, even the information that's in my brain is changing all the time, the matter. Your body is replaced every 7 years. Every molecule of your body replaces out. So all the molecules are replaced out all the time, constantly replaced. So your matter is changing all the time, but the information is the primary part. You see what I mean? The information, the code. So even if you had all of these molecules, you can't do anything because you don't have the information. So the origin of life, so say we assemble a dream team. I'll give the dream team can't even make the first cell, there's an error, the first living cell if given all the chemicals for the information code. So in other words, the mystery of the origin of life does not permit the opening of the door on a biological evolution. It's difficult to discuss biology without life. You can't start life. So say I assemble a dream team. Say we're not in caves anymore. You're in your best labs in the world, and I get the top 100 synthetic chemists, the top 100 biochemists, the top 100 evolutionary biologists, and the top 100 whatever else you want. And I give them limitless funds, and I give them all the carbohydrates, lipids, nucleic acids, and proteins that they want. And I'll even give it to them in the assembled order. All the DNA, all the RNA, I'll give you the information. You tell me the code you want. I'll give it all to you. Now you take those and just make me one cell, the simplest of living organisms, a cell. Make it for me. They'll be like, you're crazy. You can't assemble one of those. They can't. They can't. You say, well, oh, what about artificial cell? Artificial cell is you take a piece from one cell and you put it in another cell. You look what I made. It's like if I take the engine, when I used to work on cars a lot as a kid. You take the engine out of one car, you put it in another car. You say, hey, I made that car. Not, you took the engine out of one car, that's what they call, artificial cell. You try to make a cell ab initio. I'll give you all the chemicals. You can't even build it. But in some cave somewhere, it happened. Clueless. If you just Google my name, James Tour, Origin of Life, on YouTube, or just go to YouTube. Just go to YouTube and put James Tour, Origin of Life. I gave a talk at the University of Waterloo for an hour and 15 minutes. It's straight, boom, boom, just fire upon this group, just trying to explain the problems with Origin of Life. And none of my colleagues will say a word. They all act as if they've never seen it. I sent them the URL. I say, what? They don't even look me in the face because they don't want to have to address this, because they know it's a bunch of nonsense. It's nonsense what they're publishing. All right, now, the frustration of the evolutionist toward me. So now, all right, I'll give you life. What about evolution? Well, I signed a statement in 2001 that says we are skeptical of the claims for the ability of random mutation and natural selection to account for the complexity of life. Careful examination of the evidence for Darwinian evolution should be encouraged. That was sent to me in an email. Do you know how fast you go through emails? Boom, boom, boom. Yes, sure. Can you agree with this? Yes. All of a sudden, I didn't know that this was going to become the moral touchstone in court cases. But it's okay, because I do agree with it. But I didn't realize what it was going to do to my career. This statement has become known as a scientific descent from Darwinian statement. It has this amazing name now. My signing this statement has led to problems in my career. I never knew it, but it's okay, because I still do agree with it. Okay, so in 2016, just this year, so this is all new to me, I set out on a personal mission to engage biologists, philosophers of science, mathematicians, and geneticists in order to better understand evolution. I wanted to understand, what is it that you really believe? And so I have a little more access to them than a typical undergraduate student does. So I can call somebody on the phone and say, look, you're a top geneticist. This is Jim Tour. I'd like to come and visit you for a day or two. Explain to me genetics and how you get this. And they opened the door to me. Some of them were nice, some of them weren't. Some of them just berated me and just called me names. And some of them sat there very patiently and explained these things to me. So, Darwinian theory has already been debunked by the biologists. So how's that? Well, many biologists suggest that random mutation and natural selection have long been recognized by many evolutionists themselves to be insufficient to account for the complexity of life. Neutral drift is quantitatively more important than natural selection in understanding genetic differences between organisms. Furthermore, the mechanisms of evolution and their relative importance are continuously subject to careful scientific examination and revision. So careful examination of the evidence has not been avoided. So what they say to me is, you signed this statement, but we've already known random mutation and natural selection are insufficient, and we're always carefully examining. I'm like, well, guys, why don't you sign the statement with me then? Because you agree with me. Anyway, they didn't know what I was talking about. Evolution is both, this is a quote from top geneticists. Evolution is both about the mechanism by which change occurs over time and the theory of universal common descent. All right, so I sat for 2 days and just learned about common descent. It is an amazing theory, and I can understand why those fluent in the field of genetics would be convinced by universal common descent. Common descent does not mean that we evolved from monkeys, from chimpanzees. What it means is that humans and chimpanzees both come from a common ancestor. That's what common descent means. It is a remarkably well-developed theory with plenty of evidence supporting its case, plenty of mathematical evidence. We have to, as believers, do the scientific justice and be scientifically credible enough to say, I am looking at your theory and I understand why you wholeheartedly embrace that theory. It is just point upon point upon point, pointing toward universal common descent. Let's look further. So humans have 20,000 protein coding genes, which is only about 1.5% of the DNA. So only about 1.5% of our DNA is the DNA that really codes for the construction of our body. And it's within that 1.5% that common descent studies are primarily, though not exclusively, but primarily focused. Then there was a large-scale project instituted in 2003, not by Christians. This is by the U.S. National Human Genome Research Institute, called the Encyclopedia of DNA Elements, or ENCODE. You can just Google ENCODE, it'll come up. It seeks to determine the role of the remaining 98.5% of the DNA, of the genome, that was formerly poorly called junk DNA, but now it's better called intergenic regions. So 98.5% of the DNA that's within us is not what codes for most of us. It's only 1.5% that does the coding, and they thought this other 98.5% was just junk. Now they're learning it's not quite junk. There's ENCODE evidence that part or even much of the intergenic regions have regulatory elements that can affect gene transcription. That means that that's the code that builds the RNA, that then builds the enzymes that constructs us. So the uncommonness is noted in the intergenic regions, not the common 1.5% protein coding regions. If you look at the 1.5%, it absolutely looks like we have a common, that humans and other hominids come from a common descendant. When you look at the 98.5%, there's other regions there that are unique, that speak of uncommonness. Then there's work on something called orphan genes, which casts new light on the uniqueness of some genetic information. Orphan genes are considered unique to a narrow taxon, or generally a species. Therefore, orphan genes are markers of uncommonness. So in other words, you can find segments of genes where there is nothing like it that maps back to a chimpanzee. Those are called orphan genes. There are over 200 that have been recognized in human beings that are not in other hominids. That 200 are markers of uncommonness. When you look at the 1.5% of the vast majority of the protein coding genes that mark us, it really looks like common descent. But the jury is still out on the other 98.5%. The uncommon human being. Humans alone have the capacity for art, music, advanced communication, advanced mathematics, and religious practice, which constitute the broader organization of symbolism. I am here speaking to you today in symbolism. I'm describing DNA. I don't have to put DNA in front of you. I describe it, and in your mind's eye, you get it. This is symbolism. This is how humans interact in symbolism all the time. Other creatures don't do this. Try to explain to a dog, go outside, go 300 feet, and then go up a ladder, go 2 inches to the right, to the left, open up a secret gate, and you can explain that to a human every time they'll get that. Advanced communication. Therefore, if one is intent upon common descent model, there was a massive and presently unexplainable infusion, which may have been intrinsic or extrinsic, along the very short proposed pathway of descendancy between Australopithecines and modern humans. That's the thing right next to us. Right next to us. Something happened. If you look at our brains and other hominids, they are anatomically indistinguishable from our brain. You look at them chemically. They're chemically indistinguishable from our brain. We don't know what it is that makes us so much different than other hominids. Our brains are nearly indistinguishable. And the chemical mechanism, the chemical basis of the evolutionary mechanism for such changes is both unknown and presently immeasurable. We don't even know how it's different. It's clearly different. Humans deal in symbolism. If the infusion were extrinsic, in other words, if something put that in there, then the materialistic evolutionist and the design proponent share common ground. If it somehow came extrinsically. Body plans. A body plan is the ground or plan under the assemblage of morphological features shared among common members of a phylum-level group. The term is usually applied to animals, and it's the blueprint encompassing aspects such as symmetry, segmentation, limb deposition. Body plans. There is no mechanism for how different species got their body plans. No mechanism. By that I mean, remember what I told you, what the evolutionists themselves said. Biology, but that evolution consists of two things: mechanisms and universal common descent. Nobody can fathom the mechanisms for the change of a body plan in going from one species to another. Nobody has a mechanism. A mechanism means the process by which that would occur. You measure things that tell you mechanisms. Any massive functional change of a body part would require multiple concerted lines of variation. Sure, one can suggest multiple small changes ad infinitum, but a concerted requirement of multiple changes all happening in the same place at the same time in evolutionary history is impossible to chemically fathom. One day the requisite chemical basis might become apparent so that the question can be answered. But present-day biology is far from providing even a chemical proposal for the functional change, let alone a data-substantiated chemical mechanism. So I would ask them, tell me the chemical biology is not like organic chemistry where you can push every electron. Okay, fine. Tell me just in your mind how something like this could proceed. Nothing. They don't even have a proposal for the change. So, therefore, I don't understand the mechanisms needed to change body plans or the mechanisms along the descent pathway from Australopithecine brain to a modern human brain, if we were indeed commonly descended as predicted by the theory of universal common descent. I don't understand it. And nobody else understands the mechanisms either. Nobody. Nobody. They don't understand. And what the difference is, when I speak to them, I ask them, show me the mechanism. And they can't wiggle out. They're stuck. When you ask them, they will say, this is clearly understood. I will send you some papers. This is what they will say to you. Because they used to say this to me, and I say, no, I want you to explain it to me. Because I've gotten their papers, and it's a bunch of fish heads. I see nothing. I see no mechanism there. Nobody understands this. Nobody understands. My problem, I'm saying it publicly. That's my problem. Because I'm saying it publicly, that I just don't understand. I will agree that universal common descent is an amazing theory. Amazing. With a lot of backing behind it. But there are regions of uncommonness that we're seeing. And the mechanisms were clueless. And remember, evolution is about the mechanism of change and universal common descent. Recall, quoting the biologist, evolution is both about the mechanism by which change occurs over time and the theory of universal common descent, but the mechanisms are unknown, and the theory of universal common descent, though robust, is being confronted by evidence that can be interpreted as uncommonness. So further studies warranted. So we just do further studies. That's all I'm saying. Further studies are warranted. So what's the outcome of my skepticism? Was I denied tenure? No, but I got tenure 25 years ago when this wasn't an issue. None of this stuff was an issue. Loss of funding, not that I can positively identify. Harassment, not to any significant degree. Ridicule on rare occasions, but it's often, but not often directly at me. They'll say things behind my back. Confrontations, yes, but these are often diffused with just a few questions. I just ask them, okay, could you show me the mechanism? I'll give you a system that clearly evolves, and that's the immune system. The immune system evolves. It evolves and it changes, and this is how we survive. This is a microevolutionary changes. It changes. You ask them for the mechanism behind this change that we know occurs. It's right in front of us. There's no mechanism. I got one guy so upset with me, finally he sent me 70 papers. 70. He said, you read them. I mean, when somebody sends you 70 papers, it's just to try to bury you. But I set aside a weekend, and I read his papers. And I wrote back to him, I said, I didn't see any molecules in there. Where's the mechanism? You can write box, going to the next box. That's not science, that's box, going to box, one box. Where's the mechanism? Show me how it occurs. Even on something we know evolves, they don't have a mechanism. And so I just say, show me the mechanism. They move away. Have I not been hired for a position? I suspect so. Have I been excluded from professional societies? Yes. That I know has happened. And I know that because they told me, you're not getting in here because of your stance on this. So, here's the hope that I see. Science is self-correcting. If evolutionary theory is correct, the mechanistic description will become evident and the genetic studies will become more clear. In my opinion, there are many remaining questions, so further investigation is warranted. I suppose more than 99% of scientists never think about confronting anyone on these issues. They're too busy with other things. You know, most scientists are not against Christians, or they don't even think, it's a non-issue to them. I mean, you know how many emails people get? You're busy. It's busy. People never think about this. The younger generation has a deeper sense of social fairness and justice, and they're less impressed with conformal academic fluff. That's a hope that I see. That as young people start moving up into positions of influence, that they're going to say, hey, this is not right to exclude people based on their understanding of this, and they're not embracing something that you yourself can't explain. This is Richard Smalley. He won the 1996 Nobel Prize. My primary mission to which I am called is to reflect the love of Jesus Christ. Rick Smalley, 2 years before his death, came to know the Lord. We were colleagues there at Rice. And he used to ridicule Christians a lot. But then his life and his heart were softened by the infilling of the Lord Jesus Christ. What we have, the message that we have, is better than anything. The message of the resurrection. The message of the resurrection is the only thing that changes a life. Socialism doesn't do it. Communism doesn't do it. You want to change a heart, you get it filled with Jesus Christ, and the heart changes. That's what I saw with Rick Smalley. His heart changed. Max Planck said, a new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponents eventually die. I don't think that most of the opponents here are going to say, yeah, Tour, you're right. You really nailed us on this origin of life thing. It was all a bunch of garbage anyway. I don't think that's going to happen. I think they're just going to die. That's what I think. I think that the people that are embracing this are eventually going to die, and other people are going to come up and say, hey, I don't understand that either. I don't understand it. So does science make faith obsolete? Not for me.
For the invisible things of him from the creation of the world are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse.